Thank you very much for the invitation to be here. I'm deeply honored to be representing Emory tonight. So as introduced, my name is James Dahlman. I'm an associate professor in the Department of Biomedical Engineering at Emory Medical School and Georgia Tech. And when I think about today, I kind of found it a bit interesting, this title of this talk that we're sort of going through today. So does anybody actually know what the title of this talk is? It's the future of, is it health, right? The future of health. And I think we can just be honest. That's a big title, right? The future of health. And it was nice because it sort of got me thinking, right? Like tomorrow I'm giving a talk at a local university. The title of that talk is Delivering RNA Outside the Liver by Testing Thousands of Nanoparticles Directly in Vivo. So a little bit of a different vibe, right? But when I start thinking about the future of health, I realize that there are some things that, frankly, we are going to have to face that we can't predict or understand. So we are more than $34 trillion in debt, as an example, which means that every year, just to pay the interest, not the principal, it's hundreds of billions of dollars. At some point, that math adds up. That's going to affect Medicare, Medicaid, which is in turn going to affect all private insurance. It's one thing. Second thing, it takes $1 billion or more on average to get a single drug to market. It's another thing. But I am not an economist, and I'm not a policy guy. So I'm going to, for the sake of argument and sake of simplicity, I'm going to ignore all of that tonight. And I'm going to tweak the title just a little bit. All right, so we're not going to go future of health. We're not going to do that whole shebang. Instead, we're going to talk about the future of therapeutics and the future of drug development. Is that all right with everybody? All right, cool. So you might be wondering to yourself, okay, James, like, why do you feel like you can tackle even that broad of a topic, right? And I think that's a fair question. I'm going to hopefully convince you that I can at least be a credible source of some reasonable ideas about the future of drugs and drug development. And the reasons why I think I'm sort of somewhat qualified to discuss this is I have, for the last few years, been living in this world of drug development. So you guys are meeting me tonight within the context of my role as a professor. I run a research laboratory with about 20 plus students, PhD students mostly, postdocs, et cetera. A traditional academic, we publish papers, I scramble around for grants, et cetera. But there's another side to me. That's actually why I'm here in San Francisco this week. Is, as mentioned, I'm here for JPM, which is a big biotech conference. So JPM ended yesterday. If you guys had been here 2 days ago, it would have been much harder to get up here because it was packed, like sardines, packed with like every biotech executive in essentially the United States was here. So my experience as a biotech person is sort of interesting. We have started a company in Atlanta. That's not super common to start biotechs in Atlanta, but we were able to start one. It actually got acquired very quickly by one of these very cool sort of gene editing companies. There's now a second company spun out of the lab that's still in stealth mode, which means that they're not talking about it super publicly yet, but I'm quite optimistic about how that company's going to go. And we've also invented a few technologies and a few chemistries that are slowly but steadily making their way, towards the clinic. Doesn't mean they're going to get there necessarily, but it's a so far so good type of situation. Does that make sense? So again, I lecture, I teach, and all this kind of stuff. I give out PhDs or whatever, but I also have the translational side. And so that's why I think I can talk to you about this stuff, because when you think about sort of the future of medicine and drug development, what you're really talking about is this sort of weird transition from the academic center through what they call the valley of death, which is where like everything fails, into startups and then hopefully like successful startups, and those are not the same thing. So my work is in the field of mRNA therapeutics. A few years ago, you may have not heard of mRNA. As soon as COVID happened, everybody heard about mRNA. And this field developed extremely quickly. And it developed in a way that, frankly, wouldn't have been possible like 50 years ago without technology. So let's unwind that a little bit. Let's say that, God forbid, COVID had happened 50 years ago, right? Well, what's one of the first things that happened when we started seeing these reports out of, China and these places early on saying, we have, these symptoms or those symptoms? Well, the world was able to get the sequence of COVID extremely quickly. Do you guys remember this? They were able to do that because of sequencing. And for those of you in the audience who are either biotech people or scientists, I don't need to explain sequencing or why that's like exciting. But for those of you who may not be biotech people quite yet or whatever, I'll tell you that sequencing has sort of changed everything. Put simply, 50 years ago, if this had happened, it would have taken forever to figure out what was going on. And frankly, you always would have been behind, because by the time you figured out what you were fighting, it would have morphed already. Now we can sort of figure it out in real time. So I think it's a little easy for us to take that for granted. But again, not even 50 years ago, I'd even argue 30 years ago, we would have been sort of stuck, not knowing who we were fighting. So you might say, okay, that's great, James, thanks for the history lesson, but how does this stuff sort of affect the future of these things? Well, I think it gives us a nice sort of entryway into the future. So again, some of you might be familiar with a company called Grail. Grail is a company that was started that is now sort of successful, and you can go buy a Grail kit right now, do a blood draw, and from this blood draw you can say, oh, hey, we're detecting some signatures of one of 50 different cancers. So it's not a pan-cancer screen. It can't catch everything necessarily, but you can get a blood draw. That blood draw gets sequenced, and then suddenly now you can say, okay, well, there's a signature here or there. We need to be looking in the lungs or whatever. Sounds like a bit of sci-fi stuff. That is a kit that you can buy for $1,000 today. And frankly, by the time, I'm 37 right now, let's say that by the time I'm 50, 55, I would fully expect that we'll have a pan-cancer screen. So just like I go in right now and get my lipids checked, like we all do, in 15, 20 years, because of the sequencing stuff, we'll go in and we'll get a sequencing test for all known cancers. I don't think that's wild to say. All right, so that's one example of how this technology kind of comes about. All right, so now we kind of know, let's go back to COVID, we kind of know, okay, what we're fighting, so how are we going to fight it? And, we're talking about vaccines that have been administered over 1 billion times, actually many times that. And 2 of the most common vaccines were mRNA vaccines. And what we were able to do was basically take the sequence from the virus and convert that into an mRNA drug and just put it in. Some of you in the audience may have gotten your bivalent shot more recently, right? That's because we know the most recent sequence and could very, very quickly turn that into an mRNA drug. So we have this aspect of it as well. So you say, okay, again, thank you for the history lesson, but how is this going to affect the future? And this is where it starts affecting my stuff more directly. So I've been doing mRNA since before it was cool. I've been doing mRNA, I think the first time I did an mRNA experiment trying to do the very beginnings of drug development with mRNA was maybe 2011, would be my guess. So the mRNA stuff back then, again, nobody cared about it. Now people are asking kind of what's next. So what's next for mRNA is the ability, and frankly, I should say, one of its molecular cousins, which for most people in the broader world is completely unknown, but I can promise you within the scientific community is very well known. This molecular cousin of mRNA, which is called siRNA. For both of these types of drug modalities, siRNA and mRNA, we are very sort of close to developing a series of drugs. And in fact, you can go in today if you have what's called HAE, hereditary angioedema, or TTR amyloidosis. You can enroll in a clinical trial today. You can have this disease that might be running, through your family for generations and generations. You can walk in on a Tuesday, get an IV drip, and walk out without it that same day. That is already happening. So you can use mRNA to sort of encode things like CRISPR and these gene editors to knock out genes and just take them out of your genome. Again, this is not science fiction. There are clinical trials ongoing with great results published. The limitation to this technology, it almost seems kind of endless. Do you guys agree with this? It sounds like a bunch of, again, sci-fi words, but you take an mRNA to encode a CRISPR, which is a gene editor, to excise whatever gene you want out of the genome in order to treat a disease, right? Crazy, but ongoing, right? The one limitation for some of these things, for all these things, is the ability to deliver that mRNA to the right spot. So some of you might have been sort of excited in the same way that I was by the recent FDA approval of the sickle cell drugs, the CRISPR-Vertex drugs. For those of you who aren't familiar, sickle cell is a really awful disease, and it results in these very, very painful events that really make it debilitating when people live their lives. The genetics of sickle cell is actually quite complicated, but somebody designed sort of a molecular switch a while ago, and by sort of switching, or sort of by tuning this molecular switch, again using these gene editing tricks, you can basically just like, I don't want to say the word cure, but it's kind of a cure. The problem with the drug is that the treatment itself is really tough because you have to take hematopoietic stem cells out of your body, you have to edit them inside a laboratory, you then have to wipe out your immune system and then put them back in so they can engraft. This is extremely difficult. It's also expensive and frankly can be dangerous for the patient at times. So you have all these like cool molecular tools that like they're just kind of there, but you can also see this like kind of the next horizon. So today, you can go do this, what we call ex vivo treatment, take stuff out, edit it, put it back in. You can do that today for sickle, beta thal, a bunch of stuff. You can do for HAE and for TTR, you can do that IV drip thing I was telling you about. But if you are trying to work on cystic fibrosis, if you're trying to work on anything in the CNS, so Alzheimer's, FTD, ALS, Huntington's, anything like that, if you're trying to work on stuff in the kidneys, heart, et cetera, you're basically out of luck. And that is because we can't get the RNA to the right cell type. So it doesn't matter that you could, in theory, go after CF, cystic fibrosis, and like fix the mutation and correct all this stuff. It doesn't matter. Because if I can't get that mRNA into your lungs, that mRNA doesn't work in your lungs. So for the last 15 years, since the first time I picked up a pipette as a graduate student when I was 22 years old, I've been doing non-liver RNA delivery. How do I inject it with something and have it go anywhere that isn't the liver? Liver's done, we can do that already. How do you go lungs, muscle, stem cells, et cetera, And what we've been doing at Emory related to this is basically working on an idea that sort of admits how difficult this problem is. So we say, all right, I don't know how to design something to sort of skip over the liver and go to the lungs. I don't know. I mean, like I said, I've been out for 15 years, and I couldn't point to you to chemistry right now where I said, like, I can predict that this will go to the lungs. All right? So it's an educated guessing game. The way that people do this, the way that I was trained to do this when I was a student, is they say, all right, well, I don't know what's going to go to the lungs, but I can make, in my case, 2,500 different things and test them all out. Just bulldoze through stuff. So how do you do 2,500 different experiments? Well, you do them one at a time, right? So you do n equals 3 or n equals 4 per group times 2,500. Now you're at like 10,000 things. You can do that in a plastic dish. So I can make 2,500 different things. I can put them in a plastic dish, and I can test things one by one by one by one by one. The problem is, can we just like be honest for a second? Do you think that if I'm trying to inject you with something, have it avoid the liver, have it kind of avoid the heart or whatever, go to the lungs, avoid this, avoid that, you got pulsatile blood flow, you have all these organs that are all heterogeneous, you have all this stuff. Do you think that that is the same thing as a bunch of lung cells I'm spraying on in a dish? It's a problem. So you say, all right, James, we're just going to go through and do this all what we call in vivo, which is, basically adult animals, mice. But I don't think it's ethical or practical to do a 10,000 mouse experiment. So now you are stuck with a problem. How do you can make anything that you want, but you can't test it in a meaningful way. Is that clear to everybody? That's like the fundamental challenge. And so what our calling card is as a lab is basically we've been working on a way for a long time to run hundreds or thousands of experiments in one animal. And we call this barcoding. And so if you Google my name, you just barcoding. That's what's going to come up. I'm the barcoding guy, right? Like, to the extent that my professors, like my colleagues at Emory, when they make fun of me, they're like, I just barcode it. Like, that's what they just say. The way this barcoding stuff works, it's kind of a fun way to do it. Think about it, like, let's just start with like a nice, easy example. How would you do 2 experiments at once? We know how to do 1 experiment at once. Take the drug, put it into the animal, see what happens. That's 1 experiment at once. How would you do 2 at once? You might say, okay, I'm going to make 1 drug blue and 1 drug red. And then I'm going to inject them both. And I'm going to take out the lungs. I'm going to see, are the lungs red? Are they blue? Are they purple? Or are they neither? Right? Now you've done an experiment with 2 drugs at once, right? 2 experiments at once. Is that clear? Problem is you run out of colors at about 5. It's a problem. So what we did was we said, okay, 5 is better than 1, but 5 isn't important. So how can we get to 100, 200 at once? And what we did was we said we just swapped out colors for barcodes. A barcode is just a DNA sequence that you know. So we designed the barcode. We know what the sequence is. We made it. And so, experiment 1 uses barcode 1. And you set that aside, then you make thing 2 and that has barcode 2 in it. You set that aside, thing 3 has barcode 3 in it, et cetera, et cetera, et cetera. You do that 200 times, now you have 200 different things, and each one carries its own DNA sequence, and you know all the sequences. Make sense? Mix them together, put them into the animal, take out the lungs, and now suddenly you can use DNA sequencing, just like 23andMe type stuff, literally the same machine, and read out all the sequences. And so you say, okay, so you basically tell the machine, don't look for the ancestry stuff, like look for these 200 sequences. And the machine goes, oh, there's a lot of 37 in here, a lot of sequence 37. And then you say, oh, I wonder if 37 is good in the lungs. And then you can go confirm it individually. Does that make sense? Sounds very academic though, right? We would probably agree with this. Turns out it's not academic. So very early on, this idea actually worked. I bet my entire career on this idea. Entire career. It actually worked. And we started getting some attention. And what was kind of cool about this was we actually were contacted by some investors actually out here in San Francisco. I was actually having dinner with one the other day, like the original guy, the original investor. And we said, okay, like we could probably commercialize this because this delivery problem, getting things to the lungs or to the marrow or to whatever, this is a problem that everybody faces. This is a universal problem for the field. And so we were able to go start a company that we called Guide Therapeutics, based in Atlanta, an Atlanta-based biotech. And the founder was a Georgia Tech Emory PhD student named Corey. So this kid is 24 at the time, 25 at the time, basically helps me start this company, runs the company's operations day to day, and then we are very quickly, within 2 years or 2.5 years or whatever, acquired by one of the coolest companies I'm aware of, a company called Beam Therapeutics, which does all this CRISPR gene editing stuff. So just like, boom. So when we think about this stuff, I just want to emphasize that you can do the science stuff at Emory, you can do the biotech stuff at Emory, we have excellent students, and frankly, like, it's just kind of an exciting place to be. Thank you so much for your time. Thank you.